Hello, my name is Bedeir. I worked in Dr. On's lab over the course of the summer to study how the microbiome relates to overall human health, in this case specifically autism spectrum disorder. Now, the microbiome is related to a whole slew of different diseases. This includes cardiovascular disease, digestive disease, and the focus of this study, psychological diseases. What we wanted to find out is if there was any correlation between autism spectrum disorder, the presence or absence of it, and the overall microbiome. To do this, we took a data set of 246 participants divided into two groups, an experimental ASD, or autism spectrum disorder having group, and a typical development TD group. And to perform this analysis, we used a tool known as KIME2, a plugin software that generates statistical and taxonomic analyses that can overall generate plots in richness, evenness, and microbiome diversity that can help with the further analysis. What we found as results in our first plot was that the richness in the ASD groups and the both in the children and the mothers was significantly higher than it was in the typical development control groups. What this means is that the overall diversity of the bacteria in the microbiome was significantly higher in our experimental ASD groups. In plot number 2, when we plotted the microbiome structure differences across all 4 of these experimental groups, we found significant clustering in both the blue and the orange, this being the ASD and ASD mothers, compared to the TD and TD mothers, which are green and red. What this means is that there's statistically significant difference in microbiome structure among these 4 groups in that way. Finally, we wanted to analyze the relative abundance of bacterial genera across all of these different samples and across these 4 groups. After following this analysis, we found 10 specific genera to be statistically significantly different in these 4 groups, which we then further classified into 2 main columns, this being the ASD children versus the TD children, and the ASD mothers versus the TD mothers. We found 3 specific genera of note in that analysis, this being the genera Bifidobacterium and the genus Haemophilus, which are both significantly higher in ASD children and ASD mothers, and the genus Tirozella, which is significantly lower in ASD children and ASD mothers. Overall, these analyses indicate that there's significant difference in the microbiome compared to in the ASD experimental group compared to the TD control group, which may be a point for further research to help further understand how autism spectrum disorder occurs. Thank you.